this is one of 3 videos that we will be posting for your second writing assignment. In this first video, I will go over the general format that must be presented in the paper, as well as the main parts of the paper and what should be contained within each one of those sections. So we will start with the general format. So the format of the paper is going to be exactly the way you did the first one. The entire paper should be double spaced. It should be written in 12 points Time New Roman font. The paper should be presented in a third person and passive voice, and you should have no I or you in the sentences when you're talking about the experiment itself. The paper will be written in past tense as the experiments presented have already been done. And then identify each section with a bold heading to make it clear to us as a reader where you are starting and ending each section. The paper is going to start with a title page. The title page should contain a title of the experiment that you're about to present. So the title should not say just protein assay writing assignment. Instead, it should be descriptive, where you have a clearly labeled independent variable, dependent variable, and study system presented within that sentence used as your title. However, it should not be a very long title. It should be concise while presenting all the information that is needed. You should also have your name, the name of your lab instructor, as well as your lab day and time written somewhere on that title page. Your paper itself is going to start this time with the aim or purpose of your experiment. So this is the experimental question that you are trying to solve in the experiment itself. Make sure it is no longer than 1 or 2 sentences, and you are basically just talking about the main idea behind the experiment that you did, and nothing more than that. After you have presented your experimental question, you are then going to write the strategy with which you are going to actually examine your aim. Again, this section is only going to be 1 or 2 sentences. In the previous writing assignment, you wrote a detailed experimental design section. In this case, you do not need to do so. You are only presenting the main strategy that was used to test your aim or to examine your aim. Make sure that you are talking about the bread-fed assay that you are using to quantify your proteins in that strategy. Make sure the strategy is written in complete sentences and is using past tense. The next section that we are going to have, which is one of your larger section, is the results section. This section is going to be where you present all the data that you collected during class for the protein assay work. It's going to consist of 2 main parts. The first part is going to be the narrative that presents and describes your findings, and the second portion will contain the actual data that will be presented in the shape of figures or graphs or tables as needed. Now, the narrative that goes with the results is basically a text description of all the data that you are showing. Now, the best way to do that is to precede each figure, graph, or table with its related text describing what it is that you are showing underneath. That makes a logical flow of the paper and allows you to make the connections between each figure so that they flow in a story form rather than being disjointed. So in this paper, you will start by describing the main points that you are going to be presenting in the first figure, which will be your BSA standard curve, and then you will have another narrative text describing the protein quantification and the RPDs that you will do using your milk and high protein drink as your unknowns. Do not draw any conclusions in this section though. You are only reporting the data that you got and nothing more than that. So you will describe what it is that you got for your data, but you will not make any conclusions at this point. all the conclusions should be left for the discussion or the conclusion section after the results have been all laid out. So all the tables and figures must have a descriptive text above it describing the actual results that you are showing. So the first part of your results is going to be your graph. So for this lab, you will be creating a standard curve graph. This will be using a scatter plot that you will use to plot your data from the protein standard dilutions against the absorbance at 595 nanometers. Once that plot is created, you will then add a linear regression line and make sure you add the equation as well as the R-square value onto the graph before exporting it out into your document to present to us. The graph has to be formatted properly, so it should be black and white and should not contain any color. You may use grayscale if you like, but that's about the only other color that you can use. The axes, both Y and X, must be labeled with units, and you should have a title and a caption underneath the graph describing the graph above as well. Now the graph itself will look something similar to this, where you'll have your titles for the concentration of PSA mg per mil along the x-axis, the absorbance on the y, and then your y equals mx plus b equation, as well as the R-square value presented somewhere on the graph itself. Make sure the linear trend line is also showing on the graph that you export out. Once that is done, you will next present your table. Before the table, you will show the narrative text. You will have your narrative text describing the table and what will be presented within it. It should talk about the milk and the high protein mixture and the protein content that is going to be calculated, as well as that that is already known. So the y equals mx plus b equation that you are going to use to calculate the protein content is the one that you got from your own graph of the protein standard curve. That's the equation that you're going to use. Now in that equation, your y is going to be the absorbance that you got for 595 nanometer for either milk or your high protein mixture, and that is what you will substitute for the y in this equation. And x is the protein concentration, and that is what you are going to solve for from your equation. So once you have those values, those are going to be your calculated values or experimental values for the milk and high protein drink. Next, you're going to compare them to what is expressed on the label for these products itself. And that label protein content is called the theoretical protein content for those mixtures. So the information will be shown in a table form where you will contain all the information that you have calculated. However, first we have to calculate this relative percent difference to compare between the theoretical and the experimental value. It is done by subtracting your theoretical and experimental from each other, taking the absolute value of that number. You then divide that number with the average of theoretical and experimental values and multiply it by 100%. The RPD value ideally should be less than or equal to 5%. Once you have all your calculations done, you are going to present them in the form of a table, which will look something similar to this when it is properly formatted. So your first column will have your samples, which are milk and high protein drink. You will then show the absorbance at 595, the experimental concentration that was calculated using the y equals mx plus b equation, the theoretical concentration, which is what is on the label, and finally the relative percent difference. Next, you are going to have your conclusion section where you will explain your results, and this is where you will write your conclusions as well as your any differences, discuss any differences that happened. Explain why your results were the way they were and how they could have been better, how they could have made better. Finally, you will have an experimental error section, which will just discuss possible errors that could have been prevented.